Solid tumor cancers are very different than the hematologic cancers that CAR T cells have already been approved for and have shown great success in. The differences are what need to be overcome. So when people think about solid tumors, the first barrier they think of are the targets that the CAR T cells will be set for. It's easy to pick targets in hematologic malignancies because the targets are only on hematologic cells, whereas many of the targets on solid tumor cancers are on organs like liver, heart, kidney, that can also be targeted by the CAR T cells. So the first problem is finding a target that is safe. Other problems include delivery of the CAR T cells to the solid tumor, which creates about itself a microenvironment that is hostile and toxic. That's one of the reasons why solid tumors can grow in someone's body and evade the immune system. So delivery of the CAR T cells to the middle of a solid tumor is not as easy as it sounds. There are lots of competing cytokines and other lymphocyte subsets that are vying for the same tumor attention. And so a CAR T cell has a lot of competition to get where it needs to go. So we have to overcome the heterogeneity of tumor antigen expression on the surface of the solid tumor cell, and we have to be able to effectively deliver these CAR T cells to a microenvironment where they are unlikely to survive.
We need to understand what chimeric antigen receptor therapy or CAR T therapies are. Essentially, this is a way of engineering mature T cells to recognize a different antigen and includes incorporating a single strand of DNA into the genome of that T cell. That then makes the T cell to express what is a new receptor on its surface that allows it to recognize a specific antigen, and it retains the ability to signal to fully activate that T cell to engage that antigen. In order to expand this to solid tumors, we need to think about other things to make the same system work. For example, CAR therapies can target single-strand DNA, polysaccharides, polypeptides, any surface antigens. So we need to look for specific surface antigens that are probably restricted to a modified or cancerous cell. Like, for example, in head and neck cancer, we have some viral proteins, particularly E6 and E7, that are part of how the virus, the Epstein-Barr virus, modifies the head and neck cancer. That makes the cancerous cell different enough, and those can be effectively targeted with CAR therapies as we speak. However, that is just one type of cancer, and maybe the same thing can go for cervical cancer that may be driven by the human papilloma virus. But there are many other cancers. So another thing that can be done will be to look for specific genes that are only in cells that have formed cancer. There are a group that are known as cancer testis antigen, the MAGE and NY-ESO. Those are expressed by some other cancers like the gastrointestinal, melanoma, liver cancers. So if those can be similarly targeted because of the proteins that are now expressed by the cancer cell, that may be a way that CAR Ts can also be used to target those cancers.